Namaste students, my name is JVN's Dr. Neeraj Holia, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women's University, Jaipur. So today we are going to discuss a session or a topic that is molecular markers, or we can say it the genetical markers or DNA markers. So all are the same things. So, what is the need of the molecular markers? So, basically, there are conventionally a linkage map is used to map the genes on the chromosome. But with the linkage map, there are many problems which are associated. Like the linkage map is produced uh, based on the genes which produce the distinct morphological effects, and such genes are very less, or we can say that the genes which have the morphological effects are very less. So it is very difficult to map the whole genome or the correct map. Likewise, there are the mapping is tedious and time taking process using the conventional linkage maps. So there is a need of something molecular markers which uh, differences among strains at the molecular level, such as the proteins, DNA, etc. So there are many types of molecular markers are used, and broadly they are categorized either PCR based, like the RAPD, AFLP, and SSR, and the non-PCR based, like the restriction fragment length polymorphism. So in the restriction fragment length polymorphism, single restriction enzyme produces fragments of different lengths from the same stage of genome, DNA of different strains of a species or from different related species. So for to conduct the RFLP, we have to proceed the experiment step by step. First of all, we have to isolate the DNA from different species. Then the DNA are digested with the help of the selected restriction endonucleases. So the selected endonucleases, if the strains are different or the species are different, they will give different patterns on the digestion. The fragments is these digest are separated then through the electrophoresis, and the resulting gel lengths are uh, transferred to the solid support, like the nitrocellulose membrane or other uh, PVDF membrane, and then radio label appropriate with the DNA probe under the conditions favoring the DNA hybridization. So the free probes are removed by the washing step so that it will not cause the background, and the excess of the probes will be removed from there. When the fragments to which the probe has hybridized are detected by X-ray radiography or other detection techniques. So the pattern of the RFLP generated will depend on many things, like differences in the DNA of selected strain species, which restriction and endonucleases you are selecting, and the DNA probe employed for the hybridization steps. So detectable RFLP are generated due to the changes in the DNA of the organism, the changes in the base sequences of recognition site of the restriction enzyme used, relatively large deletion and the relatively large addition in the concerned stage of genomic DNA. A very large number of restriction enzymes are now available to perform the RFLP experiments. Okay, so next is the marker are the microsatellites. Microsatellites are also also behave as a marker. So the microsatellite term is given by the Jeffrey et al. In 1985. And microsatellite primers are short tandem repeats (STRs) or simple sequence repeats (SSR), which consist of 1 to 6 base pair long sequences that are repeated several times. So the number of repeats for the microsatellites is usually less than 100. The length variation for SSR is based on the differences in the number of repeats. So amplifying the tandem array and then visualizing them on a gel can detect variation in the DNA length. Microsatellite primers are mainly designed directly from the searching through the DNA sequence database for sequences containing the simple repeats. So the hybridization and sequencing are required for generating suitable microsatellite primers to study the mapping and linkage analysis in animals and plants. Hybridization using simple repeats as probes to screen the genomic clones can be used to identify the microsatellites. The flanking sequences can be obtained by sequencing the clone of fragments containing the microsatellites. Once the microsatellite markers are found, the utility of the markers is high. So DNA sequences flanking SSRs are known to be conserved in the same manner as those flanking mini satellites. These unique sequences have been used for designing the suitable primers, which are uh, polymorphic and specific for the SSR loci. When such type of primer pairs are used to amplify, they will reveal polymorphism in the form of the length variation of the amplified product. So this polymorphism is based on the differences in repeats of simple sequences. Polymorphism revealed the SSR is also termed as the simple sequence length polymorphism or SSLP. Further, these microsatellite markers use long PCR primers which are specific to a single genetic locus. They are codominant and most importantly, they are multi-allelic and detect a much higher level of DNA polymerization than any other marker system. Such simple sequence length polymorphism occur very frequently in the genome and have proven to be extremely useful as DNA markers. These properties make microsatellite primers a rich source for DNA markers, which provide specific tools for collaborative research acting as universal genetic mapping markers. Then enter simple sequence repeat markers, ISSR, and the ISR primer based on the microsatellite are utilized to amplify inter-SSR DNA sequences. It is a simple and quick method that combines most of the advantages of the SSR and amplified polymorphic DNA, RAPD. ISSR markers are mostly dominant in nature and behave like a RAPD marker. Though occasionally a few of them exhibit codominance, ISSR primers can be used variously because they can be synthesized in large number of combinations of di, tri, tetra, and pentanucleotides. Cleaved amplified polymorphic sequences CAPS markers technique is based on digestion of the monomorphic PCR product to generate polymorphism. Such digestion probes are compared for their differential migration during the electrophoresis. So CAPS primers can be synthesized based on the available sequence information from the data bank of the genomic or cDNA sequence or cloned RAPD bands. So these markers are codominant in nature, and this technique is similar to RFLP and known as the PCR RFLP. Then next marker is the EST or expressed sequence tags. So the EST markers are obtained by partially sequencing of random cDNA clones EST markers, which are synthesized from cDNA sequences, are valuable source for molecular markers and presently being used extensively. EST analysis is a simple and quick strategy to study the transcribed parts of the various genomics. So there are a number of advantages using express genes compared with anonymous sequences as genetic markers. They are useful in cloning of specific genes of interest and mapping of functional genes in various related organisms. EST are mainly used in full genome sequencing and identifying active genes which are associated with a trait of interest, and therefore EST derived markers can provide opportunities for finding a unique gene, and they can enhance the role of genetic markers by assessing variation in the transcribed and non-function genes. So EST derived markers are more highly conserved and more transferable between the species, and several genetic maps of different plant systems were constructed by using the EST derived markers. So recently, EST derived markers based genetic map was developed by the Fondvella et al. 2008 in P. So EST SSR markers are also providing highly useful in mapping of express genes and map making. So this was all about the uh, molecular markers. So hope you are satisfied with this tutorial session. If you have any query, you can write in the comment box. And this session is powered by Digital Version 2, Jyoti Vidyapeeth Women's University, Jaipur. Thank you.